Ramón, vamos a contar 5 minutos a partir de ahorita para la señal, por favor. Sí, pues que estaba esperando, pero ya está encendido. Buenas tardes. I have the pleasure to introduce Dr. Fabienne Pouline. She is our speaker for today's seminar. She is visiting us from the University of South Carolina. I'm going to mention some data of his academic biography. She is from France. She got her PhD in neurosciences at Pierre et Marie Curie University. Later, he did a successful postdoctoral in the University of Utah, Salt Lake City, in the USA. When she finished that postdoctoral position, she got a position in the University of South Carolina. Her laboratory studied the mechanisms underlying axon pathfinding and developmental degeneration in vivo. She has done interesting contributions studying how axons integrate different signals from their environment to form proper neuronal connections. And he has also done a very important contribution in the discovery of selective axon degeneration as an essential process for the formation of topographic circuits during neural development. Dr. Fabienne, please be welcome, and thank you very much for your presence.
Thank you for this introduction. Hola, everyone. It is a great pleasure and great honor to celebrate with you today the anniversary of your institute. And I would like to thank Maurizio, I don't know where he left, for inviting me to this fantastic day. So research in my lab focuses on the formation of neural circuits. And the story I would like to tell you today will show that some growing axons can actually trigger the degeneration of other elongating axons for forming proper connections. So everyone knows in this audience that your brain is your most complex organ in your body. Its functions are ensured by billions of neurons that make hundreds of billions of connections for processing information. And these connections do not form randomly during development. Instead, they are assembled into specific circuits for optimizing the transfer of information between brain regions. So if, for instance, we were measuring the activity of your brain while you are looking at this multicolor target, we would see a very specific region of your brain getting activated in a pattern that spatially mirrors what you are looking at. So this correlation, this spatial correlation between the perception of the signal and the activation of a corresponding brain region that processes this signal is enabled by the assembly of neuronal connections into what we call topographic maps. So topographic neuronal connections are assembled during development when presynaptic neurons elongate axons that navigate, sometimes over long distances, to a specific brain target where they establish contact with postsynaptic neurons. And in many circuits, these axonal connections are spatially arranged into maps such that the neighboring relationship of the neurons at the source of a signal is preserved at the level of corresponding axons at the target. So a very well-studied example of such a topographic map is the somatosensory system in the mouse, where sensory neurons that are associated with the whisker preserve their orders when they project to the thalamus, and even the thalamic axons retain that order when they project to the cortex. The visual system is another example. In that system, retinal axons maintain their neighboring relationship when they project to their brain target, except that this time the map is flipped. So you can see here, for instance, that retinal axons coming from the nasal retina, here in greenish, project to the posterior or tectum. The dorsal axons here in blue project to the ventral target. So the spatial relationship of the projection is maintained, it's inverted. And that inverted map is actually crucial for your vision because that is what allows the transmission of a perfect image of what you are looking at to the brain. So interestingly, in all these topographic systems, axons are actually already pre-ordered even before they reach their brain target. So this ordering is called pre-target axon sorting. And while it is thought to facilitate mapping at the target, it occurs independently of it. So one of the main questions in the field today is to understand how pre-target axon sorting occurs locally. And so in my lab, we're really interested in answering that question in vivo. And to do so, we use the zebrafish as a model system because it has many advantages. Zebrafish is a vertebrate that has an external fertilization, which provides large quantities of embryos. And the development of these embryos is extremely rapid. So the nervous system, for instance, is considered fully mature after just 5 days. And most importantly, you can see here that embryos are transparent. And this is great because it allows us to visualize neurons and axons thanks to the use of dye or specific transgenic animals. And finally, the genome of zebrafish has been very well characterized, so we can use many gene editing techniques, such as CRISPR-Cas9, to manipulate the genome, creating mutants or transgenic lines. So to study pre-target axon sorting, my lab studies more particularly the visual system, especially because the visual system in zebrafish is very similar to that in human. So here you're seeing two schematics in parallel. On the left, you're looking at the visual system in humans. This is a horizontal section. The eyes are at the top. And on the right is a schematic of a visual system in the zebrafish from a dorsal view with the eyes on the side. So in both systems, retinal ganglion cells, or RGCs, are the neurons in your retina that transmit visual information from the eye to the brain. And during development, RGCs elongate axons that have to go through several decision steps in both systems. So retinal axons have to elongate, have to first exit from the eye, then they elongate along the optic nerve, then they cross the midline at the chiasm, elongate along the optic tract, and finally reach their brain target, which is called the optic tectum in zebrafish. So we can visualize these RGCs and retinal axons thanks to transgenic lines that express fluorescent reporters under the control of the RGC-specific promoter ILET2B. So here is an example. You're looking at a dorsal view of a live embryo at 4 days, and you can see the RGCs labeled in green with GFP and the corresponding axons that elongate and reach their target here. So at their target, retinal axons form these maps I just introduced earlier along the anteroposterior and dorsoventral axis. Here, for instance, we are looking at the map along the anteroposterior axis thanks to the injection of specific dyes. So dye I in red has been injected in the anterior or nasal retina, dye O in green in the posterior retina. And you can see that the anterior axons project to the posterior target, whereas the posterior axons project to the anterior target. So this is the antero-posterior map, but a similar map is seen along the dorsoventral axis. So in this picture, dye D in blue has been injected in the dorsal retina, dye I in the ventral retina, and you can see on this schematic and lateral view the corresponding axons projecting to the target. Dorsal axons in blue project to the ventral part of the target, whereas axons in red project to the dorsal part. What you may notice, though, is that these dorsal and ventral axons, they are already segregated before the target in a region called the optic tract. You can see that dorsal axons elongate along the ventral branch of the tract here, whereas ventral axons in red elongate along the dorsal branch. So a big question we wanted to address was how these dorsal and ventral axons are segregated or sorted before they even reach the target. And we can think of two mechanisms for this to happen. First, dorsal and ventral axons can be sorted as they elongate along the tract during initial axon guidance. Alternatively, they could grow somehow a little bit unorganized, with some making mistake, and then the projections are corrected by a pruning mechanism that removes axons that are not at the right place. So interestingly, if we do these dye injections that I showed you earlier at an early stage, when axons are actually actively elongating along the optic tract, what you see is that the dorsal and ventral axons are not perfectly segregated at all. And in particular, there are some dorsal axons here that are shown by these yellow arrows that take the wrong path and elongate along the dorsal branch of the tract near the ventral axons here. So we can quantify this missorting by measuring the intensities of fluorescence of the blue signal and red signal along a line perpendicular to the tract at about 50 microns after axons make a caudal turn from the optic chiasm. And that would look like this. This is a trace of the intensity signals. And thanks to these intensity values, we can calculate a missorting index. So the missorting index corresponds to the intensity of the dorsal axons that are missorted, so the one on the left here, divided by the total intensity of all dorsal axons. And so if we calculate that missorting index throughout development at 48, 54, or 72 hours post-fertilization, we can first see that there is a significant number of dorsal axons that make mistakes initially. However, the missorting index decreases throughout development and eventually reaches 0 at 3 days. So what this means is that retinal axons are not perfectly segregated during initial guidance, that there are some dorsal axons that make a mistake and elongate along the dorsal branch of the tract, but somehow they are corrected. And this correction leads to proper pre-target axon sorting. So how are these missorted dorsal axons here, indicated by the yellow arrow, corrected throughout development? We can think of 3 possible mechanisms. The first one is that the missorted dorsal axons could retract and then maybe elongate back in the right place. Alternatively, they could selectively degenerate. And finally, they could translocate to the right here by a selective fasciculation with other axons. So to figure out which one of these mechanisms was involved in sorting, we repeated our dye injections, but in a living embryo. And then we monitored by time-lapse confocal microscopy of the 15 hours how these dorsal and ventral axons elongate along the tract. So the movie on the left will show the dorsal axons in blue and the ventral axons in red. And the movie on the right is showing the dorsal axons only, and the dorsal axons that are missorted and elongate at the wrong place have been highlighted in yellow for a better visualization. And so what you can see here is that first, these axons are all very dynamic. They all elongate towards their target. However, after a few hours, the ones that are missorted stop elongating and then fragment all along their length and eventually disappear. And you can really appreciate on these movies that the degeneration and fragmentation is really specific to the missorted dorsal axons. It does not affect the other dorsal axons that are nearby and took the right path, right? So using time-lapse confocal imaging allowed us to show for the first time that in the visual system, pre-target axon sorting is achieved through the selective degeneration of missorted dorsal axons. And importantly, this degeneration is specific to the dorsal axons that missort. It's uniform along their length, and it occurs before axons reach their brain target, right? So then we wondered whether somehow that degeneration was linked to apoptosis. And we wondered if p53-mediated apoptotic pathway were needed for triggering this degeneration. So we analyzed the sorting of retinal axons in a mutant embryo that lacks p53. We did our quantification of fluorescence intensities, and what you can see here is the missorting index is almost at 0 at 3 days. So this pre-target axon sorting and this pruning of the missorted dorsal axons occurs in the absence of p53. In many systems, it has been also shown that neuronal activity can trigger the pruning of synapses or axons. So we then wondered whether the activity in RGCs was necessary for pruning these missorted dorsal axons. So similarly, we analyzed the sorting of retinal axons in a mutant called macho. And these mutants actually have a mutation that leads to the complete absence of sodium channel and action potential in RGCs. And again, you can see that sorting occurs normally in this mutant, which indicates that neuronal activity is not required to trigger the degeneration of missorted dorsal axons. So to summarize on what I've told you so far, I showed you that when axons elongate along the optic tract, there are some dorsal axons that make mistake, elongate along the dorsal branch of the tract, but then become pruned or corrected. I showed you that these missorted dorsal axons actually selectively degenerate, that this degeneration does not involve p53-mediated apoptosis, and finally, that this degeneration does not require neuronal activity in RGCs. So then, an exciting question was to try to find out which cue or signal would trigger this selective degeneration. And a clue came from a mutant named DACL that was previously described as having several guidance defects in the visual system. So we analyzed pre-target axon sorting in a DACL mutant, again by injecting these dyes in the dorsal and ventral part of the retina. And we found that, like in wild type, some dorsal axons are missorted along the dorsal branch of the tract at 48 and 54 hours post-fertilization. So you can see that one here and that one here. That is completely on the left here. However, in contrast to wild type, these missorted dorsal axons do not degenerate in DACOL mutants and are maintained at a later stage, as you can see here, at 3 days post-fertilization. So we can again quantify our missorting index throughout development in a DACOL mutant. And what you can see here is that the missorting index is significant not only at 48 and 54 hours post-fertilization, but also at 72 hours post-fertilization. It remains over 10%. So this told us that in a DACOL mutant, these dorsal axons do not degenerate and are not getting pruned. And eventually, it's becoming a problem because if we analyze at a much later time of development, we can see that the missorted dorsal axons eventually reach the brain target and form ectopic connection that you can see here when we compare to a wild-type embryo, right? So what is DACOL? DACOL has been identified as a mutation in a gene called EXT2. And this gene codes for an enzyme, the EXT2 enzyme, that is responsible for synthesizing heparan sulfate. So heparan sulfate is a type of complex sugar chains that are covalently attached to core proteins named heparan sulfate proteoglycan, or HSPGs in short. And these HSPGs can be either at the plasma membrane of cells, or they can also be in the extracellular matrix. So in DACOL, the EXT2 enzyme is not functional, and this leads to the complete absence of these sugar chains at the surface of these core proteins. So thanks to these sugar chains, though, these HSPGs have many functions and modes of action during development. They can first act as receptors or co-receptors. They can also regulate the trafficking of other receptors to the plasma membrane. They can also regulate factor secretion. So for instance, FGF2 cannot be secreted by cells in the absence of heparan sulfate. They can act themselves as signaling ligand after being cleaved and released from the plasma membrane. And finally, they regulate the formation and maintenance of signaling gradients in the extracellular matrix. So overall, these HSPGs can act both in a cell autonomous manner and in a cell non-autonomous manner in the environment. So we wanted to test where heparan sulfate was required for triggering the degeneration of missorted dorsal axons. And to do that, we conducted a series of transplantation experiments using transgenic wild-type or DACOL mutant that express either TAG-RFP or EGFP under the control of the RGC-specific promoter islet 2B. So to first test whether heparan sulfate was required cell autonomously at the surface of these dorsal axons at missort, we transplanted dorsal RGCs from an islet 2B TAG-RFP donor into the dorsal retina of an islet 2B EGFP host. And the donor and host can be either wild-type or DACOL mutants. And then we analyzed later at 4 days whether these donor axons from these transplanted RGCs were missorted in the tract or not. So here, as a control experiment, you are looking at, in red here, at the dorsal axons from the transplanted RGCs. And you can see that dorsal axons from wild-type RGCs transplanted into a wild-type host navigate correctly along the ventral branch of the tract here. Conversely, and as expected, axons from DACOL RGCs that lack heparan sulfate transplanted into a DACOL host that also lacks heparan sulfate missort and elongate along the dorsal branch of the tract and then form ectopic connections. What's interesting, though, is that these dactyl axons no longer missort if they navigate into a wild-type environment. In contrast, axons from wild-type RGCs transplanted into a dactyl host strongly missort along the dorsal branch of the tract. So quantifications indicate that dorsal axons missort in dactyl into dactyl, as expected, but also wild-type into dactyl transplant, which means when heparan sulfate is lacking in the environment. And so that told us that heparan sulfate acts cell non-autonomously for triggering the degeneration of these missorted axons. So what does that mean, cell non-autonomously? It means that heparan sulfate could act in the tract environment for triggering the degeneration of these two blue axons here, or heparan sulfate could act at the surface of the ventral pioneer axons that are already present in the dorsal branch. So if this last hypothesis is correct, if heparan sulfate acts from ventral axons, we can predict that transplanting ventral RGCs that are wild-type in a dactyl environment should rescue the phenotype in a dactyl mutant. So that's exactly what we did. We transplanted ventral RGCs into the ventral retina of a dactyl mutant. And then, a few days later, we injected that blue dye in the dorsal retina of the host that is mutant to ask, are the dorsal axons of that host still missorted, or are they getting pruned thanks to the introduction of these ventral RGCs? So first, in a control experiment, we transplanted dactyl ventral RGCs into a dactyl host. The first picture here, you can see the ventral axons from the transplanted RGCs in red, and they elongate correctly along the dorsal branch of the tract. And you can see here on the picture on the right, the dorsal axons from the host that have been labeled with the dye. And you can see that, as expected, some dorsal axons take the wrong path and missort in the dactyl mutant. However, these dorsal axons no longer missort in a dactyl mutant if we transplant wild-type ventral RGCs. So quantifications indicate that this phenotype is rescued by over 50% just by transplanting wild-type ventral RGCs. So this told us that transplanting wild-type ventral RGCs was just sufficient to trigger the degeneration of these missorted dorsal axons, and that this signaling is actually a transaxonal signaling, where heparan sulfate acts at the surface of the ventral axons to trigger the degeneration of the dorsal axons that missort in the optic tract. So that was great. We were really happy to discover this, but we wondered, well, is there a specific HSPGs that does this? Because there are actually many different types of HSPGs present in the brain. And there are two main families of HSPGs. Glypicans are GPI-anchored proteins that have a big globular domain and that carry heparan sulfate chains near the plasma membrane. And syndicans are transmembrane domain that carry heparan sulfate more distally at the surface. And there are 10 glypicans and 3 syndicans that have been identified in zebrafish. So we first started by conducting an in situ hybridization, it's hard for me to say that, hybridization screen to test whether one of them was expressed at the right time and right that transaxonal signaling. And we identified one of them, glypican 3, that is strongly expressed in different regions of the brain. So here you're looking at the dorsal view of an embryo. But you may also notice some signal in the eye. And if we look more precisely in the eye, on this dissected eye, you can see that glypican-3 is selectively detected in the ventral RGC layer. So there are many different cell types in this layer, RGCs, but also glial cells. So we repeated our in situ hybridization in a mutant named Lacritz that lacks RGCs. They don't have any RGC at all. They are completely blind, these mutants. And what you can see is that we can still detect glypican-3 in the brain, but we can no longer detect glypican-3 in the retina. So this demonstrated that glypican-3 is selectively expressed by ventral RGCs. And that makes it a great candidate for mediating that transaxonal signaling between the ventral axons and the dorsal axons. So we decided to use a CRISPR-Cas9 gene editing approach to generate several glypican-3 mutants. So here is the structure of the glypican-3 gene. We targeted exon 2 and generated a mutant that is predicted to have an early truncation of the protein. We also targeted exon 7 right upstream the coding sequence of the heparan sulfate attachment site. And so we obtained a mutant that is predicted to generate a protein that contains most of the N-terminal part, but lacks the heparan sulfate. And so then we analyzed axon sorting in these mutants, and like in DACOL that lacks heparan sulfate, we found that the dorsal axons that missort are not getting pruned in the absence of glypican-3. So you can see here on this picture the dorsal axons that missort here or here, where they are not present in wild type, and this shows the quantification of the missorted dorsal axons in both mutant lines compared to wild type. So that we were really happy to see this because this told us that, combined with our in situ hybridization data, that glypican-3 at the surface of the ventral axons triggers a signal that leads to the degeneration of these dorsal axons that missort. So we then wondered whether glypican-3 was doing this by regulating another signaling pathways, right? Because I told you that all these HSPGs, they often act as co-receptors and regulating other receptors in cells. So we tested many classical guidance cues, including semaphorins, ephrins, slits, and unfortunately, none of them regulated pre-target axon sorting in the visual system. And that was super discouraging, especially for my graduate student. Let you think about that for a moment. Fail, 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 right? But then we came across a paper in the literature about a class of proteins called tenorins. So tenorins are these very big type 2 transmembrane protein that have all these very big, large extracellular domain. And one of them, tenorin 3, caught our attention because it was reported in a few papers to be expressed in the ventral retina of several species. So this is the case in zebrafish. Here you're looking at an in situ hybridization signal, and tenorin 3 is detected in the ventral part of the retina here. This is also true in the mouse, where you can see a strong expression of tenorin 3 here in green in the ventral retina. So we first confirmed that tenorin 3 was indeed expressed in ventral RGCs at the time these missorted dorsal axons degenerate. And it is the case. You can really see a strong signal just in the ventral part of the RGC layer. So we then analyzed a mutant that lacks the entire extracellular domain of tenorin 3. And this mutant was given to us by our collaborator, Robert Hinges, in King's College in London. And then my graduate student was super happy because we discovered that in the absence of tenerin 3, these missorted dorsal axons were no longer pruned. So again, they don't degenerate, and then they go to the target. So you can see that here on these pictures, and here is the quantification of the phenotype. So now we wondered then whether heparan sulfate and tenerin 3 could act together for triggering that pruning, right? And to test that, we use what we call a non-allelic non-complementation assay. So a non-allelic non-complementation assay tests whether an embryo that is heterozygous for 2 mutations has a phenotype, whereas heterozygous for one of these 2 mutations has no phenotype. So if you have gene 1 heterozygous from gene 1, no phenotype, heterozygous for gene 2, no phenotype, you analyze a double heterozygous for gene 1 and gene 2. And if you don't see a phenotype, you can pretty much assume that gene 1 and gene 2 act independently to exert their physiological effect. However, if you see a phenotype, that tells you that gene 1 and gene 2 act in the same pathway for exerting their effect. So we analyze the sorting of retinal axons in DACL heterozygous, tenerin 3 heterozygous, and DACL tenerin 3 double heterozygous. And what you can see on this graph is that we can clearly see a phenotype in a double heterozygous mutant, but not in single heterozygous mutant. And this is even more striking when we analyze the number of missorted fascicles, also the number of dorsal axons that missort and are not getting pruned. We can detect a lot of them in the double heterozygous, but not in single heterozygous. So all these data combined tell us that tenerin 3 and heparan sulfate and most likely glypican 3 act together at the surface of these ventral axons to initiate a signal that leads to the degeneration of these missorted blue dorsal axons. So if they both act on ventral axons, we then wondered, well, what could be the receptor at the surface of the dorsal axons that receives the signal? So very recently, there are several papers by several groups that have shown that tenerins are able to bind in trans to a class of adhesion GPCR receptors called latrophiline. And these trans interactions between tenerins and latrophilins are mostly known to promote the formation and stabilization of excitatory synapses. So they have a really important role in that regard. So we wondered whether a latrophilin could be involved in our system, and we identified 6 latrophilins in zebrafish. But one of them, latrophilin 3.1, was strongly expressed throughout the RGC layer at the time missorted dorsal axons degenerate. So we then analyzed a mutant that is predicted to have or generate a truncated form of the receptor. So the extracellular domain are still present, but the entire GPCR is lacking, transmembrane and intracellular domain. And what you can see is that we can detect the presence of missorted dorsal axons in about 35% of these mutants. So all that combined, that told us that glypican 3 and tenerin 3 at the surface of ventral axons initiate a pruning signal, probably by activating latrophilin 3, which we assume would be at the surface of the dorsal axons, considering the known trans interactions between tenerins and latrophilins. So we were really happy to have figured that out, but one last question we wanted to address was whether there was a critical time window for this to happen. Can these dorsal axons be pruned all the time, or is there a very specific time period during which that can happen? And so to address this question, we decided to use a transgenic rescue approach in the DACOL mutant that lacks heparan sulfate. So we generated a DACOL mutant line that also expresses a transgene, and that transgene consists of a wild-type version of EXT2, the enzyme that is mutated in DACOL, under the control of a heat shock promoter. So that means that the transgene is only expressed if we apply a heat shock on embryos. So if we cross a DACOL heterozygous that is transgenic for this rescue construct with the DACOL heads, we obtain a clutch of embryos in which 25% of embryos are mutant, and among these mutants, 50% are going to have the rescue transgene, and 50% are not going to have the rescue transgene and can be used as control. So we then perform a heat shock at different time periods of development to induce the expression of EXT2 at different time. And then we wait and we inject dye in the dorsal retina to analyze whether the dorsal axons are still can be pruned or not when we re-express EXT2, right? So in the absence of heat shock, so just the mutant, we can see that about 80% of the mutants have missorted dorsal axons. And this is true for the ones that don't have the transgene and for the ones that have the rescue transgene. So now if we perform a heat shock before axons reach the optic tract, we completely rescue the phenotype as expected. So you can see here that we perform a heat shock at 30 or 34 hours post-fertilization. So at that stage, retinal axons are just arriving at the chiasm. They are not in the tract yet. And you can see that the transgenic mutants that are in black here do not have any missorted dorsal axons, which means that these dorsal axons were pruned. What's interesting is when we heat shock the embryos after axons have elongated along the tract. So here, 48, 56, 64 hours post-fertilization. So, but that time, dorsal axons have already elongated in the tract, and yet we can still rescue the phenotype. So that means that reintroducing the presence of heparan sulfate at a late stage can still rescue and trigger the degeneration of the dorsal axons that missort. However, if we perform heat shock even later, at 72 and 80 hours post-fertilization in particular, we can no longer rescue the phenotype, and the DACOL mutant have missorted dorsal axons, whether they have the transgene or not. So this told us that there is a specific time window during which reintroducing heparan sulfate in that mutant rescues the phenotype. And what we think is happening is that during that time window, dorsal axons and the signaling I showed you can still effectively prune the axons. But if we wait too late, these dorsal axons reach the target, form synapses at the target, and then are stabilized. And then it's too late. We can no longer get rid of them. So altogether, all the data I showed you today demonstrates that pre-target axon sorting in the visual system is established through a transaxonal signaling between ventral axons and the dorsal axons that missort along them in the dorsal branch of the tract. This transaxonal signaling must occur during a specific time window before these dorsal axons are stabilized at the target. And this transaxonal signaling is initiated by glypican-3 and tenerin-3 at the surface of ventral axons that likely activate latrophiline-3 at the surface of these missorted dorsal axons. In response to this signaling, these missorted axons degenerate, and that leads to proper pre-target organization of the axons for facilitating mapping at the target. So with that, I would like to thank the people in my lab who conducted the work. Dr. Olivia Speed was my former graduate student. She conducted all the studies of glypican-3 and heparan sulfate. Dr. Corey Weaver was my former postdoc, and Trevor Morland is my current graduate student, and they've conducted the studies on tenerines and latrophilins. I would like to thank our collaborators, especially Robert Hinges, who gave us the tenerine-3 mutant. We are recruiting graduate student, postdoc as well, so please reach out if you're interested. And I would like to thank you for your attention, and I'm happy to take any questions.
Hello. Thank you for your fantastic talk.
Thank you.
I have two questions. The first one may be too simple or maybe naive, but don't target cells have any say whether to distinguish if an axon was misguided or not?
So you mean at the target?
Yes.
We don't think so, because there is very early work that was done in the 70s and the 80s in which they were really, really good then, because they removed the target. They managed to remove the tissue that would become the target from the embryo, and then they analyzed the sorting of axons, and it occurred normally. So we really think it's completely independent of the target.
Okay, that's very clear. Thank you. The second question is, in polysynaptic pathways where, say, somatotropy or retinotropy is maintained, is it the same mechanism in between all these steps in the polysynaptic pathways?
You mean in other systems, for instance?
Yes, for example, in the visual pathway, there is a synapse in the lateral geniculate, and then there is more axons that have to spread all the way to V1, and they retain their retinotropy. So is the same process equal for every step of the way?
This is a really interesting question. Short answer, we don't really know, because it's actually very difficult to study pre-target axon sorting in the mouse. So they know that pre-target sorting occurs, but they cannot, nobody has tested whether degeneration occurs, because they did that on fixed tissue. And so if you fix the animal after there is nothing left, you cannot see anything, and before, then you don't see what happens to missorted axons. So we do not know if in mammals or in the rodents, if it's the same mechanism or not.
Okay, thank you very much.
What about the timing of the different axons? Are they all going at the same time? The timing?
The what?
The timing when the axons
the timing.
Are they all going at the same time exactly?
No, so that's a very interesting question too. Thanks for bringing that up. The ventral axons actually elongate first.
Yeah, that's what I thought. Yeah.
And I think they serve as a reference then for the other dorsal axons.
Hi, thank you. First, when the dorsal axons are, the ventral axon, no, dorsal axons. When the dorsal axons are first extending, is it entirely random, or is there already a bias toward the favored side?
So we do not know with the technique we have now if it's random. By using this dye injection, we can tell that about 25% of the dorsal axons take the wrong path. Now we don't know if they just randomly make a mistake or if it's a subpopulation of dorsal axons. So that we don't know. But we're favoring the random aspect, because in other systems, for instance at the chiasm, it has been shown that there are random errors that are made and corrected. So we think that it's also a random process here, but it's hard to prove.
Thank you. And also, for the ventral axons that are expressing these molecules, how do they know, is there an asymmetrical expression, or how do they know whether an axon is on the wrong side or the right side?
So that's a great question, and it's actually something we thought a lot about, because what puzzled us was, why do they even grow in the wrong place initially, right? Because initially they elongate here, and they are not getting pruned, and they're totally fine, and then all of a sudden they fragment. So that means that these dorsal axons become sensitive to a signal. They are not sensitive initially, and then they become sensitive. And what we found, and I'm not showing here because it's really preliminary, but what we found is that latrophiline and tenerin 3, they're expressed really early on, from the very beginning of eye formation. However, glypican-3 is not expressed right away. And so when these dorsal axons elongate first, glypican-3 is actually not there. And we see a correlation between the onset of glypican-3 expression and the gain of sensitivity of these dorsal axons. So what we think is happening is that the expression of glypican-3 sets the timing of degeneration. And then, to go back to your question of which side, the big difference that we see between the left, where they missort, and the right, is that these missorted dorsal axons really have a direct contact with the ventral axons. Whereas these guys, they're a little bit separated, and they're not in such a close contact, and we think that's the difference. Yeah. Sorry, it was a long answer, but...
Yeah, well, one of my questions was about that, whether there is also a time frame expression of the receptor. So
yes.
If this is similar to this critical period that you are detecting.
So we think the culprit is glypican-3. But I'm speculating now. We are in the process of testing that, of trying to make new lines to test whether if we force glypican-3, if we force the expression of glypican-3 earlier, can we even prevent this missorting or trigger the degeneration earlier?
So the receptor is present the whole time?
Sorry?
The receptor is is there the whole time?
Is
okay.
Based on in situ hybridization. So the mRNA is there the whole time. We assume the protein follows the mRNA, but unfortunately we don't have any antibodies to test the protein yet.
Yeah, and I wonder whether this can apply to other neurons.
Yes, that's a good question.
Independently of the phenotype of the neurons. So these neurons are cholinergic?
The RGCs?
Yes.
Glutamatergic encephalopathy.
Glutamatergic, okay. So whether the dopaminergic or cholinergic neurons are also showing these mechanisms?
Yeah, there are a lot of work that has been done in Drosophila in particular, where they show pruning and degeneration of axons during metamorphosis, for instance, when there is a rewiring of the circuits, and it's really interesting to see that it seems to really involve specific pathways that can affect multiple classes of neurons. Yeah.
Thank you.
Mm-hmm.
More questions or comments? Thank you very much.
Thank you, everyone.